Lisa. Wait, is it pointer? All right, thanks, Morten.
Thanks, Martin. So sounds good. Thank you. Thank you very much for the invitation for me to come here today and present to you. I have a mixed bag of different things from a company perspective. So yeah, I'll just get started and then we'll take the questions in the end. My name is Morten Danielsen. I'm the manager of MSomics. MSomics is based in Copenhagen. It's 9 years old now. It's a service provider of metabolomics services. So we take in samples from research projects and analyze them. And it's like microbiome metabolomics. We do a lot of fermentation samples. It could be preclinical samples or human samples. So a vast array of samples. And a little advert here to begin with. We will soon be posting some positions, so I certainly hope to hear from many of you, of course. I have with me also, we have Rosa on the team today, down in the corner waving. So if you want to know how it's like to work at MSomics and not just hear me say how wonderful it is, then you can talk to Rosa. She's here during the course all week. So we started as a small startup, 2 people, and in 2018, 5 years ago, we moved to a small site north of Copenhagen. If you know the technical university, then we're just a few kilometers north of that. And we have been lucky and been able to expand ever since. So we did new labs in the last few years. And the coming maybe this year or next year, we will also start a lab in the US. So that will also be part of the work maybe for a new scientist to be involved in. Now, so I come from MSomics, but actually MSomics was acquired by a larger company called Clinical Microbiolomics. We still keep the 2 names. Clinical Microbiolomics is a major genomics company, a bioinformatics company. So now we cover both DNA, major genomics and metabolomics, and multi-omics data integration in the company. And Rosa is actually an example of this collaboration. She worked at Clinical Microbiolomics before and was now transferred to MSomics doing metabolomics now. So coming back to the topic on quality assurance. So I have a few of the company slides here. First, also about the environment that we work in. So we have like a European Medicines Agency or US Department of Health. They have some guidelines how to work also. So when we started in 2014, small company, like we get 20 samples from different scientists to work on, analyze these, and then make a living out of that. But now, as we're growing, now it's 1,000 samples, it's 3,000 samples, it's big pharma, big biotech companies coming to us to have these investigated, and they want us to have a really, really high quality standard. Just like you saw when you visited the biobank, it's really important to document and be able to tell that you have this quality. And when you're doing metabolomics, it can be quite difficult because metabolomics is a new technology. So there are not many established guidelines. There's not many strict protocols on how to do it. You might find a white paper describing, best practices or these guidelines, but it's still new and evolving. So what we deliver to client and what we have is data, and a lot of data. You saw on the biobank side that they have like thousands and thousands of samples, or even millions of samples. We don't have that much samples, but we generate a lot of data from them during the metabolomics, and how to manage that and how to ensure that we have the right data and have the right quality for the clients is of course extremely important. So when this is also like a nice company slide presenting how to build sort of the pyramid of quality and end up with a validated method and be standardized, even so that we want to be in a position where you can have a clinical study and you can have endpoints, and these endpoints could be metabolomic endpoints that you're measuring. But how do you come from having samples and going to that? It's a long journey, and it starts by many of the things that Martin already alluded to about the quality control and how you build up that. But one thing is also the part where you build the stuff that Martin was talking about, that you have all the right procedures in place, you have even documented it, but then you build on that, and then you make sure that you can document that all your staff has been trained in all the methods, they understand the method, they document the methods when they do it, and when you train new people, they will deliver the same results as the people who train them. So there's even when you have the guidelines or have the subs, then you have a lot of work to do before you're in a complete GXP fashion or working, for example. So this is the way we present how we work, but this is no more different from your work probably. You have the study planning, and then you have where you have the samples, you need the collection storage. You do extraction, data generation, data analysis, and then the final reporting. What's a little bit different for us is that we don't do the collection and storage, and sometimes we don't do the study planning. So we only brought into the picture when the samples are ready to be analyzed. But in order to comply with everything, then we need to be in place already in the study planning, so we can make sure that we deliver in the end what is needed, for example, if it's a clinical study. But it also means that our business is changing from getting samples with a few weeks of notice to now being part of planning clinical studies, where sometimes we will get samples. Now we're talking about getting samples in 2025 for studies we're planning now. So it's a whole other time frame also to be working on. Let's skip this one, but different things to consider. We discussed also, so I'll bring up a little bit about GDPR also. GDPR. So GDPR is the General Data Protection Regulation that you know when you click on the cookies and on your computer and have to consent with everything, and the foundation and background for this is to give everybody control and right over their personal information. So this is of course extremely important. Just like we had questions in the biobank, do they ask people before your samples are used in a study? So that could be considered like part of the data protection about your samples and how you share data. What it means for us also as a company is that even if we get, say you have a study, 50 samples, you say, okay, I label them 1 to 50, then we can send to MSMX. There's no problem with GDPR because they don't know what 1 to 50 is. But according to GDPR, because you have the translation table from the samples 1 to 50 to who those persons are, then the samples are not anonymized. They're called pseudonymized, which means that someone will have a code that can translate it, meaning that we have to live up to GDPR rules, because in theory, someone will get the code and translate it. We even have someone claiming that because we get samples that might contain DNA, then it can be linked to people, and therefore it's not an anonymous sample. This means that we have to act like a data processor, meaning we have to know who accesses the samples, meaning our freezers, It has to be in a lock room. We need to know who's accessing the samples. We have to log access to computers. Not only do they have to have passwords, we need to make sure that people are not trying and forgetting their password, because then we have to report that someone has tried to log in without remembering their passwords. You have to have two-factor identification. For samples collected in Denmark, we have discussions, can you store at Dropbox, even though we have an agreement with Dropbox that they only store on European servers? That's not enough. So for those samples, we handle them specifically and only store them locally. And we have to store them in locations where not every employee can access them. Before, we had the system where everybody could access everything, to make sure that, if someone is sick or ill, and then we can make sure that the studies keep on moving. But now we have to include these layers of extra data protection. So it of course means that we are better living up to GDPR, but it also means that it's more difficult to work, but that's just the way it is. Yes. So a little bit about the project workflow, the study planning. That is when we come into study and we want to discuss with clients what is it that they need, and then they can choose between the different methods that we have. It could be amino acids or fatty acids, bile acids. It could also be broader, untargeted metabolomics methods. The main question we ask here when we do the planning, is it a standard project where we can put in subs and we know that everybody in the lab would know how to work with it? Or is it a customized project? It might be that we need one of our senior scientists to work on this, because it's could be mouse brain samples where we have a few milligrams of each sample, and you need to make sure that the scientist really knows what's going on with these samples, because they cannot be replaced if we make mistakes. We have to make sure that before we get samples, and I would also recommend this to every one of you, if you're working in a lab facility where you have collaboration with partners or people coming from outside, write down what you have agreed you would do to the samples. That removes a lot of issues afterwards, if there's any issues with the samples or with the data. And make sure that the client or your partner understands the limitations of what you're doing. If you say you're going to measure amino acids, but there are maybe two of them missing, or one of them is not very good with the method, make sure that the client understands this, so that they are fully aware of this in the beginning, and can tell you if it's hugely important for them to have a specific amino acid measured in high quality. Then we have more like legal stuff that are a binding contract and stuff about how we do it, and this is also part of the work. You need good sample descriptions, of course, but sometimes it's not as simple. Like you saw the box of mixed samples. Sometimes we feel that this is the way some clients would be sending samples to us, and then we can just figure out how to analyze them, or how to make sure that the old blood samples have a high quality. So we try to have the conversation, of course, before we receive the samples, to make sure that people know actually what they're sending to us and what is needed in order to deliver high quality metabolomics data. And also that we get, for example, we get the sample list, it's filled in by the client, so that we don't make mistakes by typing in what is on a 9-digit label when we get the samples. So here, yeah. So these slides. Sorry about all the animations coming here, but we have you see the sample list on the left, and the intention of the slide was that when you have the samples, when you process the samples, you can have really good subs that describes everything that you need to do. But sometimes it's also really good to have an experienced scientist looking at the samples, say, okay, if they don't precipitate like they should do, or if somehow the samples turns brown and you expected it to be yellow, what's happening in the samples? And often it's really good to have these experienced professors or senior scientists around, and not just blindly following standard procedures just because you believe these are validated. So even when you try to write down everything and have the highest quality possible, you might be able to do it for, you know, high throughput blood samples, but when you get tissue samples, mouse samples, all kinds of different samples, then also common sense and experience is really, really good to have in the lab. Then we have the thing about the instruments, and we run on 4 or 5 different platforms. We have 2 different kinds of orbit traps. We have the Bruker timsTOF, and then we have GC instruments where we run amino acids and short chain fatty acids, and also both liquid injection and headspace injection. So all these instruments also need, of course, protocols and maintenance, cleaning and calibration procedures. And like the slide Martin was showing, you know, when you have a drop in the QC, what does this mean? Is that a method where you might sometimes experience this, and it's not important because what you're looking for is in the samples. You can see even if you have a small drop in the QC in the beginning, or is it a method where you know every time the QC needs to be exactly like this, or your SST or your other controls. So for these, we have 9 instruments in total, and we have 1 person employed just mainly for keeping the instruments running. So it's also a major task of running the instruments. It has also, for us, it's also been part of this going from a small startup with 2 people to now we're close to 20 people working on metabolomics. But now we have 1 person maintaining the instruments. Before, a few years ago, every scientist maintained and cleaned the instrument before running, but now they can trust that all the instruments are running smoothly when they have the samples ready. And then we use the vendor of the instruments to do and come in and check the instruments annually. Then a little bit about the data analysis and study reporting. The data workflow we have is very similar to also what Martin described. We have the raw data, and then we have a small part where we make sure that we do that immediately after the sample are running, or if it's a batch running over several days, we do the check every day to make sure that we can stop the samples and restart them if we need to do that. Because if you wait until the data scientist is ready 3 weeks later, then you might need to rerun everything, or instead of just starting and stopping the instrument again. So also when the instruments are running over the weekend, typically a scientist will just log in and check that everything is good. We use Skyline and Parafact2 and Compound Discoverer and the Bruker software that I forgot what's called for the peak integration. And then we have different databases also for peak identification. And then a little bit about how we use the data for normalization and the analysis. So for the quality control of the data is similar to Martin's description, but I'll just show you how it looks when we're doing it in Skyline, for example. So first of all, the retention time is checked, and that you don't have variation over the retention time. We know for each method how much variation we maximum can have. Of course, we want the internal standard detected in all samples, and it seems obvious, but it's not always that people check it, but it's a simple check to make sure that you actually have injected sample into the instrument and that it's measured. And then you have about how much variation would you like to see as maximum for the internal standard. And this also depends on the instrument and what you're running on. Then we look at if we have targeted compounds that we look at specifically, we also do the same. In this case, we also use the mixed pool quality control sample, and in this case, the variation is limited to a maximum 20% within the QC. So we check the QCs, and if we have a large run, it might be like 10, 20, or 30 QCs, and you don't want a high variation between the QCs. You check the linearity and the range of the calibration curves, if you have included these, and then the variation of your measurements. And this is a bit how it looks like in when we do it in Skyline. It's a software developed for proteomics. I don't know if you're going to look at it. No, I see shaking on the head, but it's a nice software to check, and it's a freeware also. It was developed for proteomics, but it works well for metabolomics too. Compound Discoverer is a thermal scientific software, so that's comes with the instrument, so that's definitely not freeware. And when we do untargeted methods, so then we combine the data processing in 2 steps. We use the Skyline for the targeted detection of compounds, and then we use Compound Discoverer for the untargeted method or detection. I'm not showing it here, and I don't have a slide on it with me here, but when you use a targeted method, then we're able to go lower into the noise of the instruments and make sure that this is actually the compound we're looking for. With the Compound Discoverer, with the untargeted, we go a little bit higher in threshold to make sure that we don't include too much noise when we analyze the data. And then we have the results that are matched to internal and external libraries, and then we annotate them based on how confident are we on the results, so that also clients get a clear idea whether we know what we're talking about, or can they trust that at level 1 annotation is very good, and how about level 3 annotations, what can we do with that? So we typically also have client meetings where we talk about, how can they interpret the data, which of the data should they use first of all, and how can they build their results and their understanding of what's going on in their studies. Yeah, and this is similar to what Martin was saying, that sometimes it's needed that you normalize the data, and we often use the QCs to normalize also, or use the internal standards to normalize also. How are we doing on time? 5 minutes. Okay, I have a few examples of study designs that were not perfect, or at least that influenced the data, and then we can take some questions afterwards. So, for example, often clients or projects are sample limited, so that you don't have as much samples as you would like, or you need to distribute your samples between different experiments. So a question we very often get is, how much sample do you need? And if you ask the lab technician or lab scientist, they would like a lot, so they can repeat the study and use a lot. But sometimes you can't do that. So, for example, if we look at serum or plasma from mice, then we can go down to 20 microliter or even less. And then what you see on it's not a really good PCA plot, but what it shows, if we go into the details, is that we've done a study where you test different amounts of tissue, and when does it start to affect the results of the study. And in this case, we could go down to 15 milligrams of tissue before we begin to see that the variation between the sample extractions influence the results. This is an example where you have around 80 mice samples, but because they needed 80 mice samples and they didn't have that many mice at the same time, they needed to split it into 3 cohorts. And even though the study was really well balanced with all groups represented in each cohort, then when we do the first data analysis, what you clearly see is that the cohort, it influences the results. And we have analyzed all the samples together and processed them all together. So the variation comes from the different cohorts of the mice. So you need in the data treatment then to correct for for the different cohorts, for example. Another example is here that you have 8 cages of mice, and you see this kind of PCA plot, and it looks not perfect, but fairly okay with separation of many of the groups. But when you then look closer, then you see that one of the untreated controls are mixed with the treatment controls, and this we could see because we had the cage effect. Otherwise, these 6 samples would have been mixed together. A study where they collected many samples, and one of the primary results was also that they didn't make one big batch of plasma tubes. They had 2 batches of plasma tubes, and you could easily tell the difference between the plasma tubes that they have used. So you also needed to correct for this in the results. And this is another example of where you see that, first of all, you see differences between sampling sites when we did the analysis. And then finally, the last slide, that sometimes you think you have a very good design also, but there can also be problems like you have too large a difference between your study groups. So that if you want to investigate a specific effect of a drug, for example, but then when you do metabolomics, you have 500 significant molecules, then the difference between the study is maybe too big to find out how is it actually working if you have smaller doses or and so what's often seen, and what many people have pointed to, that this is also that sometimes you have too small study groups, or you have too many confounders like age, sex, disease states, like was alluded to before by Martin, also that how you do the design, but it can also be that you simply don't have enough samples, or you want to investigate too many things in the same study with too few samples. So therefore, now you're the scientist doing the analysis, but try to get into the discussion on the study as early as possible in the study, so that you can inform people, how you're doing the analysis and what is needed in order for them to have high quality results back when you have had a look at the data. And the final part is that sometimes we have also seen this, that people want to have the results fast, so they ship half the samples to you, and then the second half afterwards when you have analyzed the first batch. And that's not a good idea when you know that you get batch differences. Or you can also assume that, well, maybe the samples were left without being frozen immediately, and then you assume that they're fine, but maybe we should check that before we're running a big study. Or that we assume that when you say you measure amino acids, it also means that you measure amino acids derivatives or other special things. So always first most have a good discussion with your project manager or the ones sending samples or data to them. That was all I had, and I'll be happy to answer any questions.
Any questions?
Yes. Well, the question was, what do you do if you don't solubilize all your sample? And I would say it again comes back maybe to the question you have about what you want to analyze. But if it's something near that you want to compare groups, and some groups are solubilized and other groups are not, or that you want to do some kind of quantification, then you need to think about your extraction method rather than assuming that it will be okay. Yeah.
Thank you, Morten. And now there's a coffee break and then some cake and non-cake alternatives. And we will be back 4 to 4. 12 minutes.
Right.